Hi guys, so welcome back to my channel. So today we are going to go into subtopic 1.2 still under Form 5 Kssm Chapter 1 Organization of Plant Tissue and Growth. Okay, so the subtopic for today will be meristematic tissues and growth. Now previously you have learned about types of tissues, correct? We have the permanent tissues and also meristematic. So today we will go deeper into meristematic. So I hope you are ready with your pen and paper and take notes, as much notes as you can. All right, and yeah, let's do this. Okay, as usual, we have to look through the learning standard before we go in, so you know what you are supposed to learn. Now, here, what is your learning standard? The first one is to identify the types and parts of tissue involved in growth. Okay, so what are the one involved in growth? And then describe the position of zone of cell division, zone of cell elongation, and zone of cell differentiation in shoot and root. So this is the growth zones, the three types of zones related to growth, which you are going to see later. And also you are supposed to describe the types of growth. We have primary growth and secondary growth, and then justify the necessity of primary growth and also secondary growth. Why is it so important? And then to compare and contrast the primary growth and the secondary growth, the similarities and differences in eudicotyledon plants. Okay, and then to assess the economic importance, okay, of plants that have undergone secondary growth, basically the importance, economic importance of trees, kayu bala, kayu meranti, and that kind of things. Okay. Okay, now let's look at meristematic tissues. Now, what is meristematic tissue? It is actually tissues that are made up of actively dividing cells that have not undergone any differentiation. Okay, the cells have thin cell wall, they have large nucleus, dense cytoplasm, and also no vacuum. So in the previous class, you also have seen the types of meristematic tissue, which is the apical meristem and also the lateral meristem. So the apical meristems, they are always growing, so they are at the root and at the shoot. Okay, always growing, they never ever stop growing. Okay, and lateral is the one that grows sideways. Okay, so up and down apical, left and right lateral. Okay, so yeah, so there are two types of meristematic tissues. Now so we have here apical meristematic tissues, okay, causes primary growth, okay, meaning lengthening of the plant. They grow taller, okay, basically, okay, and it occurs at the tips of shoot and roots, and it produces new leaves and flowers. So basically, that's the one for it. But lateral meristem causes secondary growth. They grow sideways, okay, that means the wood becomes bigger and bigger. Okay, it occurs at the cambium. Where is the cambium? Oops. Let me erase this for you so you can have a look. Where is the cambium? So we have two. One is vascular cambium, one is the cork cambium. Okay, and lateral meristem also produces bark on trees. Okay, so if you look at this picture here and this picture over here, don't get scared. This is actually the apical meristem here, and this is the root. Okay, apical meristem of the root, which are always growing. Okay. Okay. Now let's go into the zone of cell growth. Okay, so we have zone of cell division, zone of cell elongation, and zone of cell differentiation. How do we remember? Well, division, so we just remember divided. So it starts with dividing, division. Okay, and then we have the elongation and also differentiation. Okay, so divide first, elongate, and differentiate. Differentiate means it changes its shape or function to perform, specific function. Okay, so we have this tree here. Okay, now let's look at the division. Now, primary growth leads to an increase. Okay, increase in length of the plant shoot and root. It is due to the activity of the apical meristem. So it's always growing. Okay. And then we have here the growth phase, the cell division. Okay, in this zone, the apical meristem in the shoot tip and also in the root tip will divide actively through mitosis. Okay, you have learned mitosis in the last year, Form 4. Okay, so you know what is mitosis. Now, the meristem cells are very small in size, okay, with large nucleus and several small vacuoles and also thin cell walls. Okay, if you can look at your textbook, you can see that picture over there. Now, as new cells are being formed, the cells which have been formed early are pushed to the zone of cell elongation. Okay, I will show you more pictures. Now, in the zone of elongation here, or we say enlargement, so in plants, usually they increase in size, okay, due to the absorption of water into the vacuoles by osmosis. Okay, and this process is actually called as vacuolation. Okay, so this causes several small vacuoles, okay, small vacuoles to actually enlarge, okay, and then finally they fuse together to form one large vacuole. Okay, so from small vacuoles to one large vacuole, and the hydrostatic pressure, okay, that acts on the cell wall will cause the cell to enlarge until the maximum size is achieved. Okay, and at elongation phase, the growth rate is usually the fastest. Okay, and then we have the zone of cell differentiation. Okay, when after the cell has reached its maximum size, okay, the usual plant cell with a large vacuole, so the cell differentiation will take place to form permanent tissues. Okay, and the cell will begin to change its shape, okay, and structure to form specialized cells with specific function. Okay, and in plants, usually the epidermal cells in the roots will differentiate to form root hairs, okay, in order to absorb water from the soil. And other cells also differentiate to form phloem, xylem, okay, mesophyll, guard cells, pith, and the cortex cells. Okay, so if you look at the picture, this is the differentiation between xylem and phloem. Yeah, so this is for the roots, for the shoot. Okay, if you look here, we have zone of cell division. Now, this is the shoot apical meristem. Okay, then we have here, this is number 1. Number 2 here is the zone of cell division. Then we have zone of cell elongation, where the vacuole becomes enlarged. Okay, and the size, goes to maximum capacity. And then we have the zone of cell differentiation. It differentiates into xylem, into phloem, cortex, epidermis, okay, guard cells, and so on, whatever cells you have in a plant. And once they have achieved, everything has been differentiated, they are called as mature tissue. They don't undergo anything anymore. Finish, that's it. Okay, so this one, if you look at this is for the shoot. Okay, so we have 1, 2, 3, 4, 5. So don't get confused with the zone. Okay, most students, they like to put this is the zone of cell division. No, this whole thing is zone of cell division. Okay, then comes in elongation, differentiation, and mature tissue. Okay, I'll put another picture for you. Yeah, so this one is root. Okay, if you look at this, same thing. Okay, same thing. Now we have here, this is the root apical meristem. Okay, this one, this is root cap. This is root cap. Don't get confused. Although it is the outermost here, this is not the apical. This is the root cap. Apical is here. Okay, this is apical. Root apical meristem. Okay, then we have, this is number 1 here, zone of cell division. Number 2 will be zone of cell elongation. Number 3 here is zone of differentiation. And number 4 here is mature tissues. Okay, so that's how you remember. Okay, just remember divided. Divide, elongate, and differentiate. And then they become mature. They are grown up. Okay. Okay, now, types of growth. Like animals, plants also experience growth, correct? So most plants continue to grow throughout their lives. All right, so in animals, growth takes place all over its body. And on the other hand, in plants, growth is only concentrated on specific parts such as meristem. Okay, so there are 2 types of growth in plants. We have primary growth and also secondary growth. Now, primary growth occurs at the tip of the shoot and root. Okay, basically the apical meristem. And it occurs throughout the year. Whereas secondary growth occurs after primary growth is completed. And this one occurs in the stem and also roots. And it involves the lateral meristem. Actually, they grow wider. So only woody plants and shrub undergoes secondary growth. Herbaceous and monocotyledons, they only undergo primary growth. Okay, now let's go into the details. Okay, now primary growth. Now this growth takes place in the apical meristem at the shoot tips and also root tips. Okay, so primary growth starts when the meristem cells in the zone of cell division, okay, the cell division, zone of cell division of the apical meristems are actively dividing. Okay, so this will be followed by, after they divide, they elongate and they differentiate, okay, into other cells. Now, at the shoot tips, okay, the leaf primordia, okay, this is the leaf primordia, okay, and the shoot primordia, okay, will grow to form new leaves and shoots. Okay, so this enables the plants to increase in height. At the root tips, okay, the root cap become exhausted. Okay, so then, you know, they penetrate the soil, so they become exhausted. Okay, so this causes the cells of the root cap to be replaced by the meristem cells. Okay, so that is primary growth, usually at the shoot and the root, the apical meristems. Okay, so this is the leaf primordia, okay, which is always growing. Okay, if you look at this, leaf primordia, here, around here, okay, we also have at the roots. Okay, now secondary growth. Now, as the plant grows taller, it will need to increase in diameter or in the girth, G-I-R-T-H. Okay, I'll write it down for you here. Okay, diameter, increase in diameter or girth, or the plant may break easily. Okay, so secondary growth refers to an increase in the circumference or diameter of the stems or roots. Okay, so it usually occurs in woody plants such as teak, oak, okay, forest tree, fruit trees, and also dicot plants. Okay, but they are rare in monocots or monocotyledon plants. Okay, now secondary growth is the result of cell division in the lateral meristem. Go sideways, huh? Okay, and both the stems and the roots. Okay, both stems and roots, they undergo secondary growth. Now, lateral meristems are the vascular cambium and also the cork cambium. Okay, if you look at here, we have the vascular cambium. Vascular cambium is around the vascular bundle. Okay, in between the xylem and phloem, and then they form one whole. Yeah, here they form the whole cambium. Okay, cork cambium is around the cortex. Now, yes, okay. Next picture. Yeah, if you look at here, okay, so you can see, we have the cork cambium here and also the vascular cambium. Okay, vascular cambium located between the primary phloem and the primary xylem of the vascular bundle, and the cork cambium is located immediately below the epidermis. Okay. Okay, now let's look at the secondary growth at the stem. Now, secondary growth in dicot involves the activities of the vascular cambium and also the cork cambium. Now, let's look at vascular cambium first. Now, the cells in the cambium ring will divide inwards to form xylem and outwards to form phloem. Now, if you look at the stem cells, okay, this is the vascular bundles. Okay, this is the vascular bundle. It looks like flower inside here. Okay, and then it's divided into xylem. This is xylem. Okay, and this outer one will be the phloem. Okay, here xylem, this is phloem. Okay, so this is xylem inside, phloem outside. So now what happens is the cell in this cambium ring is around the vascular bundle. Okay, so it goes this way. This is the vascular ring. Okay, which is in between the xylem and the phloem. Now the cells in this cambium ring will divide inwards. Okay, inwards to form new xylem and outwards to form new phloem. Okay, the new xylem, okay, the new xylem tissues will become secondary xylem, and the new phloem tissues will become secondary phloem. So as a result, the primary xylem is pushed towards the pith, while the primary phloem is pushed towards the epidermis. Okay, pith is in the middle. Okay, this is the pith. So xylems will keep moving towards the pith, and the phloem will keep moving towards the epidermis. All right. Now, the walls of the secondary xylem is thickened with lignin. Okay, lignin to give mechanical support to the plant. And the addition of all secondary tissues causes the epidermis to be stretched and split. Now, activities of the cork cambium. Okay, what happens here? Now, the cells of the cork cambium, which is below the epidermis, will divide, okay, actively to form an outer layer of cork cells and also inner secondary cortex. Now, the walls of the cork cells contain a waxy substance called suberin. Okay, so this suberin will provide an impermeable layer to water, and also it will take the role of the epidermis. So now we don't have epidermis, we have this cork cambium. Okay, so this cork layer forms a protective bark, okay, bark, which is actually known as the peridome, okay, on the surface of the woody stems. So this bark consists of cork and cork cambium and also phloem. Okay, so if you look at this is the whole thing. Okay, the peridome has layer peridome, the bark, and the cork. Okay, then followed by the division of the cork cambium, and then followed by this is the vascular cambium. Okay, I'll show you in the next slide, much clearer from your textbook. Okay, so this cork actually will replace the epidermis if the epidermis split, and being waterproof, it prevents the stem from losing water and protect it from mechanical injury. Okay, so if you look at this is the stem, how the stem looks like. Okay, this is primary growth. Okay, we have, this is the xylem, this is the phloem, and in between is the vascular cambium. Okay, then it grows inwards. So what happens is, this is your secondary xylem, and this is your secondary phloem. So the primary phloem pushed towards the epidermis, the primary xylem will be pushed towards the pith. Okay, and then the cork cambium, when they actively divide, they form bark. This is the whole bark, including the cork, okay, cortex, and also the phloem, primary phloem, okay, and secondary phloem. Where is secondary phloem? Yes, secondary phloem. Okay. Okay, so this is the picture from your textbook. Same thing. Okay, it is the same thing actually. Okay, so secondary growth starts when the vascular cambium divides actively through mitosis, and the cells in the cambium ring will divide inwards to form new xylem, outwards to form new phloem. So remember, in xylem, out phloem. Okay, and then the new xylem will become secondary xylem. The new phloem becomes secondary phloem. So it's all basically the same thing. It's a much clearer picture from your textbook, page 11. Okay, so you can go through that. All right. Okay, now let's look at the secondary growth at the root. Now, secondary growth in roots is similar to the secondary growth in the stem. Now, secondary growth takes place in mature roots. Okay, the vascular cambium is also located in between the xylem and the phloem. Okay, the vascular cambium. Now, so what happens is that it divides laterally, okay, to form ring of vascular cambium. Now, the layer of the cambium cells, okay, the cambium cells will divide to produce secondary xylem on the inner side and the secondary phloem on the outer. Same concept, okay, in xylem, out phloem, okay. And then what happens? The root then will increase in thickness, okay, due to the activity of the vascular cambium. Okay, then there is also pericycle. Okay, there will be a pericycle here which divides to form the ring of cork cambium. Now the cork cambium divides to produce cork cells that protect the tissues of the root. Okay, so we have here same thing. Look at the primary xylem, you look at the primary phloem. Okay, wrong spelling here. All right, and then we have the secondary phloem and also the secondary xylem. Okay, primary xylem goes into the pith, and the primary phloem towards the epidermis. So you can look at this at the end product, secondary phloem here. This is the vascular cambium. Okay, then we have the secondary xylem, the pink, light pink, and in the middle one is your primary xylem. And lastly, we have the cortex here. Okay, so this is secondary growth in roots. Okay, now let's look at annual growth rate. How do we get the age of these trees? Okay, so in temperate countries, the activity of vascular cambium is not uniform throughout the year. Okay, so in spring and summer, the cambium is more active and it forms larger and thinner wall xylem vessel, okay, because of the favorable conditions. And lighter rings are formed in the stems. In autumn and winter, okay, when water is limited, the cambium is less active. So it forms a very narrow vessels and also fibers. That means small cells, but with thicker cell walls. Only a few secondary xylem cells are produced. So hence, we have the darker rings, okay, formed on the stems. Okay, and then the wood, okay, that's formed in the rings in the spring is called spring wood. Okay, and while the wood from the autumn is called as autumn wood. So this kind of wood are seen together in transverse section of the stem, okay, as concentric rings called annual growth rings. Okay, so each annual ring indicates 1 year's growth. So by counting the total number of annual rings, the age of the tree can be determined. But trees in tropical countries like Malaysia, okay, the trees here, they do not show clear annual rings because there are no seasonal climates. Okay, so we can check. Now, if we look at another picture here, so from here you can count the rings. Okay, so this tree is actually 24 years old. Okay, so we have 1, 2, 3, 4, and so on. Okay, so that's how you count. Okay, now let's look at the secondary growth of monocots. Okay, most monocots are very small and herbaceous, so they are actually generally lacking of secondary growth, but there are certain exceptions. Okay, for example, palms, where they produce a wide procambium region. Okay, that means they have a large diameter base with many strong vascular bundles and much vascular parenchymas. Okay, so they are not that strong compared to the ones that really undergo the secondary growth. Okay, but they have that so that they can give support to the plant. For examples, we have here the coconut trees, okay, rice, banana, pineapples, and corn plants. Okay, so these are some of the examples of monocots. Okay, usually they don't undergo secondary growth except for a few. Okay, now the necessity of primary and secondary growth. Primary growth is very important for cell elongation. Okay, and then the primary xylem and primary phloem is also important to carry out its specific function. Xylems, we know that to transport water and minerals, and then we have also to support the herbaceous and young plants. Okay, so that it doesn't it doesn't fall off. Okay, not being flexible and all that. So it needs the xylem, and phloem is to transport all the nutrients, the products of photosynthesis. Okay, so they need that. Now, secondary growth is important is that so that the plant, okay, is stable throughout its life. Okay, it provides stability, it provides mechanical support. Okay, it produces more xylem and phloem so that more water and nutrients can be transported throughout the plants. As you are growing, you need more nutrients. Correct or not? Even you, when you are growing, you need more nutrients. Same goes with plant. And then secondary growth is also important to replace old tissues. Okay, so they replace old tissues, and then they can produce a stronger and thicker bark, okay, to prevent a microorganism attack, okay, or any other thing, weather kind of thing, and also so that the plants can live longer. Okay, so that's why secondary growth is also very important. Okay, done. Now, this is a question from summative practice 1. Okay, so this is a longitudinal cross section of the shoot tip. Okay, here they say shoot tip. Yeah, if you look at this, it looks like a devil. This is definitely the shoot tip. Okay, now name zone 1, 2, and 3. So we have zone 1, okay, zone 2, and zone 3. So what is it? Zone 1 will be the, remember, divided, D, E, D, divide. Okay, so we have what is that zone? Now differences. What is the difference between zone 1 and zone 2? Okay, now then they ask you to draw a cross section of the shoot at X and Y. Okay, that is the mature tissues. Okay, so what is that? And after a few years, okay, the plant undergoes secondary growth, and draw a cross section of the plant stem which has undergone the secondary growth. Now you can do this first question, okay, and then come back and check your answers. Okay, so here we know the divided just now. Okay, zone 1 will be zone of cell division, 2 will be elongation, and 3 will be cell differentiation. Now, what's the difference between zone 1 and zone 2? Yeah, okay, the cells in zone 1 have larger nucleus compared to the nucleus found in cells from zone 2. And most cells in zone 1 have small vacuoles, while cells in zone 2 have large vacuoles. Okay, so you draw that two things, the plant cells in zone 1 and also in zone 2, the vacuole we are talking about, and also the nucleus. Now, cross-section of shoot at xy, yeah? So we cut it there. So what we get is this one. This is a stem. Huh? So we get, if you look at this picture, this is the one we get. And the D, the question says, cross-section of plant stem which undergoes secondary growth. They want plant stem. So again, drawing the stem. This is for primary growth. This is after secondary growth. That means you have the secondary xylem and also secondary phloem. Okay? Okay, so then you have to compare between primary growth and also secondary growth in eudicots. Okay, now why do we need to compare? Of course, we need to compare. We want to know what is primary growth and secondary growth. Okay, so this is one of your learning standards. So this is the example given in your textbook. You can copy this. Okay, copy this down and study this well. You know that in primary growth, apical meristem is involved. Secondary growth is the lateral meristem. Okay, and then which part of the plants that undergo the growth? Okay, which is the direction? Okay, longitudinally or radically, up, down, left, right. Okay, and then what are the tissues and structures formed? Okay, presence of woody tissue. Primary, no woody tissue, but secondary we have woody tissues. Thickness of bark. Primary growth, very thin bark, but secondary growth, thick bark. Okay, and then presence of annual growth ring. So growth ring usually present in secondary growth. Okay, and both growths occur in woody plants. Okay, primary and secondary. Okay, and it involves division by mitosis. Okay, so you can go study this table. Okay, and make your notes. Okay, this is also another bio exploration that you can see in your textbook. Okay. Okay, now let's look at the economic importance of plants that have undergone secondary growth. Now we have many kinds of timbers. Okay, but the most famous ones that we have in Malaysia, we have the shorea, which is the meranti, and the balanocarpus, which is the cengal. Okay, jati. There are other kinds of timber as well. Now, what are they so important? Why are they so important? Because they are used as structures for houseboats, furniture, okay, fences, doors, buildings, okay, all needs wood. Okay, so and they are also used as decorative items, okay, commercialized as varnish, adhesive substances, perfume, and also medicine. And the fruits are also commercialized. So, timber production is actually giving a lot of benefits to the economy. Okay, so you can go through this info for the examples of secondary plants in Malaysia. Okay, and also to further study on the types and uses of timber in Malaysia. Okay, and present the information in the form of brochure. So I have one example for you. I'll show you. All right, so this is one of the brochure that is done by my students. So you can have a look at this. Okay, how do you prepare a brochure? Okay, timbers in Malaysia, what are timbers? Okay, and the importance and the types of timbers. So type of timbers we have here, rubber wood, okay, meranti, nyato, and also teak. Okay, so what is it important for? And also the importance of timber, okay, as fuel source, okay, and then for what is it called, furnitures, and also paper manufacturing. So yeah, so that's why we need all this kind of timber. So there are actually more. Okay, if you do your research, you can get more. All right. Okay, done. We are done. So you know your learning standard. As long as you cover your learning standard, you know what is you are supposed to do. You can answer this question. Okay, formative practice 1.2. Okay, name the 3 zones of cell growth. Very easy. Discussed. Okay, now number 2, they says figures below shows a cross section of plant stem that undergoes secondary growth. Label primary xylem, primary phloem, secondary xylem, secondary phloem. You can refer this to your textbook or the slides that I have been showing you. It's there. Formation of secondaries. Explain the formation of secondary xylem and secondary phloem, growing inwards, growing outwards. Done. Now, why is secondary growth important to plants? Also covered in the slide. Now, secondary growth supports primary growth. Using your knowledge in biology, support the above statement. Yeah, because the primary growth, okay, that will actually grow into secondary to give more stability, to give mechanical support. So how? Okay, explain it. Done. Okay, so done. We are done with all this. So I hope this video has helped you a lot. Do give me comments if it doesn't. If you have any other questions, we can always discuss this in the comment box below. Okay, so yeah, with that I will end my video here. Thank you for watching, and please do not forget to like and subscribe my channel. Bye bye.